Hey everyone, ready for a deep dive? We got a request to unpack some dense research, and it's brain tumors, specifically canine and human meningiomas. Those tumors in the meninges, right? The protective layers around the brain and spinal cord.
Yes, exactly.
And get this, they're the most common primary intracranial tumor in both humans, and you guessed it, our canine companions.
Fascinating, isn't it?
It is, and we've got some really cutting-edge studies here, so I'm super excited to break it all down with you today.
I'm happy to be here. I think the parallels between human and canine meningiomas, it really gives us a unique opportunity to understand this complex disease.
Okay, so let's just get right into it. I understand that not all meningiomas are created equal, especially in humans. Can you tell us a little about the different subtypes?
Absolutely. So in humans, we have three distinct molecular subtypes: Mengi-A, Mengi-B, and Meng-C. Now, these classifications are based on differences in gene expression, DNA methylation patterns, and chromosomal alterations.
Whoa, that sounds pretty complex.
It is. But basically, Meng-D and B, they're generally considered less aggressive, but Meng-C, that's a different story. It tends to be much more aggressive and often recurs even after treatment.
So are you saying there isn't a good treatment for Meng-C?
Well, the real challenge is that we currently lack effective treatments specifically for Meng-C.
Meng-C sounds like a formidable opponent. What makes it so aggressive at the molecular level?
So Meng-C tumors, they have four key hallmarks that contribute to their aggressive nature. First, they display chromosomal instability, meaning their DNA is prone to errors and rearrangements.
Okay, that makes sense.
Second, Meng-C tumors have a much higher rate of cell proliferation, meaning those cells are multiplying rapidly.
So it's growing quickly.
Yes, exactly. Third, we see an upregulation of the DREAMM-B complex and its target genes. This complex regulates the cell cycle, and its overactivity in Meng-C just fuels uncontrolled growth.
Okay, I think I'm following.
And finally, Meng-C tumors often exhibit promoter hypermethylation, a process that can silence tumor suppressor genes. This further contributes to uncontrolled proliferation.
So Meng-C is like a runaway train, with multiple systems failing. No wonder it's so hard to treat. But taking a step back, I imagine studying meningiomas, any subtype, is challenging.
Oh, you're absolutely right. Researching meningiomas, especially in humans, it faces hurdles because of the limitations with current models. Primary cell cultures become senescent quickly, they age and lose their ability to divide.
I see. That makes them not as useful.
Yeah, and another approach is to create patient-derived intracranial xenografts, but these are really difficult to establish and maintain.
So if studying human meningiomas directly is so difficult, where do our canine companions come in?
This is where it gets interesting. This research explores the possibility of using canine meningiomas as a model to understand the disease in humans.
And why dogs?
Meningiomas are really common in dogs, and histologically, they're very similar to human meningiomas.
So by studying these tumors in dogs, we might unlock clues about how to fight the disease in humans. Maybe even Meng-C.
Exactly. And that's the main question this study is trying to answer. Is there a canine counterpart to that aggressive human Meng-C subtype?
So how did they even start to answer that?
Well, they dove deep into the molecular makeup of canine meningiomas.
I hope it was more than just a couple samples.
Oh, yeah. They analyzed 62 canine meningiomas from 27 different dog breeds.
So a good amount of diversity.
Yes. And they used RNA sequencing to analyze the tumor samples.
For those of us who don't live and breathe molecular biology, what is RNA sequencing and why is it so powerful?
So imagine you want to understand how a cell works. RNA sequencing lets us take a snapshot of all the genes actively being transcribed into RNA at that moment. It's like listening to the conversations within a cell.
So RNA sequencing is like eavesdropping on the tumor cells.
Exactly. It reveals which genes are switched on, which are switched off, and to what extent.
Wow, so much information. How do you even analyze all of that data?
It's definitely a lot of data. That's where bioinformatics comes in. They used methods like principal component analysis, or PCA, and consensus clustering.
Those are some pretty technical terms. Can you break those down for us?
Of course. Think of PCA as simplifying complex data, finding the main themes in a huge library.
Okay, I get that.
And consensus clustering is like grouping books together based on shared themes.
So PCA helps us see the big picture, and consensus clustering reveals the details.
You got it.
What did these techniques tell us about canine meningiomas?
They revealed something remarkable. Within the canine meningiomas, they found three distinct molecular classes, mirroring the human subtypes: Meng, A, B, and C.
So just like in humans, wow. Does that mean one of the canine classes acts like Meng-C?
That's what we all want to know, right? But there's a twist. Remember Merlin, the protein made by the NF2 gene? Its loss often drives meningiomas in humans.
Yeah, you mentioned it before.
Well, here's the surprise. They didn't find any significant losses on chromosome 26, where that NF2 gene is located, in any of the canine tumors.
So the loss of Merlin, which is so important in humans, might not be a major player in canine tumors.
Right. It suggests that Merlin loss might not be as critical in canine meningiomas. This actually lines up with some recent human studies, too.
Interesting. So less about Merlin. What about that aggressive class in canines, the potential Meng-C mirror? Did they find anything similar to human Meng-C?
They did, and it's really interesting. So remember the 4 hallmarks of human Meng-C we talked about?
Chromosomal instability, increased cell proliferation, dream complex upregulation, and promoter hypermethylation.
Yes. This study focused on the first 2, chromosomal instability and cell proliferation.
Got it. Let's start with chromosomal instability. What did they find in the canine tumors?
So they saw something interesting. Histologically, some tumors in class 3, which we think might be like Meng-C, showed anaplasia.
Anaplasia. That doesn't sound good.
It basically means the cells are losing their normal structure and becoming more primitive.
A sign of aggressive tumors.
Yes, exactly. And to look closer at chromosomal instability, they use an algorithm called Casper.
Like the friendly ghost.
Well, this Casper is a bit different. It assesses large-scale chromosomal gains and losses from RNA sequencing data.
Ah, so it can spot chunks of DNA that are either duplicated or missing.
Exactly.
And what did Casper reveal?
It showed that class 3 canine meningiomas did have significant chromosomal instability, just like human Meng-C.
Okay, so one hallmark down. What about cell proliferation? Were those class 3 tumors dividing rapidly?
They were. They looked at the expression of MKI67, a marker found in the nucleus of dividing cells.
The more MKI67, the faster the cells are multiplying.
Exactly. And guess what? Class 3 canine meningiomas had significantly increased MKI67 expression compared to the other 2 classes.
So those class 3 tumors are dividing quickly, just like Meng-C in humans. 2 hallmarks down. But before we move on to those other 2 hallmarks, I have a question.
What is it?
We keep talking about the similarities between canine class 3 and human Meng-C, but are there any key differences?
That's a great question. While the similarities are striking, we're still comparing different species. There are bound to be differences.
Of course, that makes sense.
We need more research to really understand those nuances.
All right, so back to those remaining 2 hallmarks of Meng-C. What about the upregulation of the dream complex and its targets? Anything similar in canine class 3?
They did, and this is where comparative genomics is so powerful. They used gene set enrichment analysis, or GSEA. Think of it as a way to see if groups of genes, like those in a specific pathway, are overrepresented in your data.
So they're looking for patterns of gene expression rather than individual genes.
Precisely. And the GSEA results were really clear. They found a significant overlap between canine class 3 and human Meng-C, specifically elevated expression of cell cycle genes and upregulation of dream target genes.
Wait, so canine class 3 tumors also have that dream complex overdrive?
They do. And the specific genes involved are remarkably similar between the 2 species. Genes like FOXM1, MYBL2, CCND1, CDK1, BUB1, TOP2A, COL6A3, and that MKI67 we talked about.
Wow, so even the individual genes are similar between canine and human aggressive meningiomas. That's incredible. It really shows how we can use canine meningiomas as a model for this disease.
I agree. Now, this study didn't look at promoter hypermethylation.
Right, it's just one piece of a big puzzle.
Exactly. There's still much more to explore.
Absolutely. But this study found so much already. Now, our listeners probably wondering about what this means for dogs with class 3 tumors. What's their prognosis?
Well, that's the burning question. Unfortunately, this particular study doesn't have a definitive answer. Managing canine meningiomas is more variable than in humans. It often comes down to the owner's choice and resources, but they did have some follow-up data for some dogs in class 2 and 3 who had surgery.
What did it show?
It's limited data, but there was a trend towards shorter survival times after surgery for dogs in class 3 compared to class 2. More research with more dogs and longer follow-up is needed to confirm this.
That makes sense. It's still early days in this research, but the potential is huge.
Absolutely. And that's what makes this study so groundbreaking. It's the largest transcriptional analysis of canine meningiomas to date. It confirms the existence of molecular subtypes, and it identifies an aggressive class similar to human Meng-C.
It's like we've been handed this treasure map, and we're just starting to figure it out. Who knew our furry friends could hold so many clues about fighting human brain tumors? This is amazing.
It really is. This is a major step forward for understanding meningiomas in both species.
Okay, but before we get too ahead of ourselves, we need to pause our deep dive here for now.
Stay tuned for part 2, where we dig even deeper into these fascinating tumors and explore the potential treatments this research has uncovered.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.
Welcome back to our deep dive, into this fascinating canine and human meningiomas.
We left off with some pretty big news. We learned there's a canine class of meningiomas with some crazy molecular similarities to that aggressive human Mengele subtype.
Right. It really is remarkable how similar they are.
So can we use these findings to actually develop better treatments?
Well, that's the million-dollar question, isn't it? We aren't at the point of, directly using this in humans, but this research opens up some really exciting new paths to explore.
Like what? I'm very curious.
Well, canine class 3 tumors, they have a lot of the same hallmarks as human Mengele, so they could be incredibly useful as a model for preclinical drug testing.
So if a drug works in a dog with a class 3 tumor, it's more likely to work in a human with Mengele, kind of like a stepping stone to human trials.
Yeah, exactly.
Makes sense. So we could test new drugs in dogs, and then if they work, move on to human trials.
That's the idea. Of course, there are ethical considerations and regulations we have to work with.
Right, of course.
But the potential is definitely there.
That's good news, especially for anyone facing Mengele. You mentioned preclinical drug testing. Are there specific drugs or targets that researchers are looking at based on all this?
There are a few promising things. Remember that Dream complex is upregulated in both canine class 3 and human Mengele tumors.
Yeah, it's like that master regulator of the cell cycle.
Right, and it's stuck in overdrive, leading to all that uncontrolled cell growth.
Got it.
So one thing we could do is develop drugs that can actually inhibit that Dream complex, or its downstream targets, basically hitting the brakes on that runaway cell cycle.
Stop the tumor from growing so fast.
Exactly.
Are there any drugs that can target the Dream complex in development right now?
There are a few early-stage compounds that are showing some promise, but it's really early. The problem is the Dream complex is involved in a lot of important cellular processes. So if we inhibit it completely, it could have side effects.
All about that balance, right? Killing the tumor without hurting the patient.
Precisely. That's where precision medicine is so important. Developing drugs that can target the tumor without hurting healthy cells.
Using what we learned from this study about the genetic and molecular changes in these tumors.
Exactly. That's how we can start developing more targeted and effective therapies.
This study looked at a pretty small number of canine meningiomas, though. What's next in terms of research? What do we need to do to confirm all this?
We need larger studies with more dogs. It's a good start, but we need more data to confirm all this and really explore the possibilities.
More dogs, more data, more insights.
And it's not just about the number of dogs. We also need really good data, detailed clinical information.
Like their medical history.
Yes. Tumor location, imaging data, how they were treated, and long-term follow-up.
To see how they do over time.
Exactly. That way we can connect the molecular findings with what actually happens to the dog and figure out what makes some tumors more aggressive than others.
Like risk factors.
Exactly.
And we also need data about the owners and their dog's environment too, right?
That's right. Things like exposure to pesticides, household chemicals, that kind of thing, even breed predispositions.
So we can learn if those things play a role.
Right. And that could even help us understand the human side of things too.
So we learn from our dogs, and that helps us understand and treat the disease in humans.
It's a two-way street. We're all in this together.
Exactly.
This study really shows how connected human and veterinary medicine are.
Totally. We've learned from our dogs, and it can actually help us humans too.
Exactly. It's a win-win.
Okay, but we have to pause our deep dive again for now.
But stay tuned for part 3, where we'll finish up our exploration of canine and human meningiomas.
And we'll talk about the big picture implications of all this.
It's going to be good.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging. All right, welcome back to our deep dive. We've covered so much ground already, from the molecular details of these tumors to the potential for new treatments.
Yeah, we've been through a lot, haven't we? But it's been fascinating.
It really has. And, throughout our deep dive, we've seen this incredible connection between human and veterinary medicine. It's like a reminder that we're all in this together, humans, animals, the whole planet.
Absolutely. One health, right. What affects one of us affects all of us.
Exactly. One health. And this study It really embodies that principle.
Absolutely.
Right.
By learning about canine meningiomas, we're not just helping dogs, we're potentially helping humans too.
It's like we're all connected, even at the cellular level.
That's a good way to put it.
And, by studying both canine and human meningiomas, we get a much more complete picture of what's going on.
Totally. It's like having two pieces of a puzzle that fit together to make a whole.
And that knowledge can then be used to develop better treatments for both species.
And that's what comparative oncology is all about, right? Studying cancer across species.
Exactly. And this study highlights just how important that is.
It challenges our assumptions about how these tumors work, and it gives us new directions to explore.
Speaking of new directions, this study has the potential to make a real difference in people's lives.
Absolutely. We've uncovered some crucial similarities between canine class 3 and human Meng-C meningiomas, and that could lead to new treatments, new hope.
It's amazing to think that our furry friends could be the key to finding a cure for this aggressive form of brain tumor.
It is, and this is just the beginning.
That's right. There's still so much more to learn.
But this research gives us a solid foundation to build on.
It's exciting to think about what the future holds.
I agree, and I think this study serves as a powerful reminder of why we do what we do.
To help animals, to help humans, to improve lives.
Exactly. And to remind everyone that we're all in this together, working towards a healthier future.
Well said. And on that note, I think it's time to wrap up our deep dive.
It's been a pleasure, as always.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.